Welcome to MOOC course on Introduction to Proteogenomics. This is Dr. Bing Zhang's hands-on session on WebGestalt. WebGestalt or Web-based Gene Set Analysis Toolkit is a functional enrichment analysis web tool. Dr. Bing Zhang will briefly describe about the software and its 3 well established and complementary methods for enrichment analysis, including over-representation analysis ORA, gene set enrichment analysis GSEA and network topology based analysis NTA. He will also discuss about different parameters of each method, input gene list and run the analysis. So, let us welcome Dr. Bing Zhang for today's session.
I am going to introduce 2 tools that have been developed in my lab, mostly for pathway and network based analysis, and also for using these tools to analyze data that has been generated by TCGA and the CPTech cancer programs. So one of the tools is called WebGestalt, the other one is called Proteogenomics. So I think I'll give a little bit of structure for this hands-on session. So first I want to give a very brief introduction to the WebGestalt tool, and then we can have some practice, and then we can have a little bit break if you want, or we can go to the Proteogenomics introduction and the hands-on part. Because I think the underlying method has been described both in some of the Carson's talk and also my talk yesterday, so the introduction can go very quickly. So for the WebGestalt, so this actually was my first bioinformatics project, and it was a tool I created when I was a postdoc. So the motivation is that when you do RNA-seq or proteomics and these high throughput technologies, actually when I started this I was doing microarray at that time. And then usually you need to do like differential expression analysis and clustering analysis, this type of things, right? And eventually you end up with a list of genes that are of possible interest. Like a list of differentially expressed genes, or through clustering analysis introduced by Dr. Manley during the previous lectures. You are going to get clusters of genes that have similar expression pattern, and you want to explore those genes. One way to do this is to leverage the information we already have about the pathways. So and the typical path, first, in order to do this, you have to have some pathway definition, right? And like the pathway databases, or actually it can be also like loosely defined functional categories or just any gene sets. And then there are 2 types of analysis: over-representation analysis and the gene set enrichment analysis. I think we already talked about this yesterday in Carson's lectures. So in WebGestalt, so we have collected a lot of pathways, functional categories, and other types of gene sets. Basically it's from different databases or through some computational analysis. I won't, because it's too small, I don't think you will be able to see it, but maybe you have the lecture note, so you should be able to read on your computer. But it can be separated into different categories. And in total, there is a very large number of gene sets that can be used for your analysis. For the overrepresentation analysis, just a quick reminder, you have a list of genes of interest. Let's say you do an RNA-seq or proteomics experiment, and then you get a list of differentially expressed genes. And then you want to compare with a gene set or a pathway group to see whether there is any association between them. For example, we want to compare this with the development of gene ontology, and then you can do the overlap, and then you can count the number of overlapping genes, right? If you see any overlaps, that indicates some of your genes are involved in this biological process. But the question is whether this is enriched representation of that category or not. You don't know, right? And then what you can do is to randomly sample the same number of genes as you can from next 581 genes, randomly sampled from the proteome you studied, for example, and then you can do the overlap. And for example, here you observe the 98 gene overlapping here, but only 65 in a random experiment. And then you can say, okay, I have more overlapping genes than here. But is that significant or not? In order to do that, you do the Fisher's exact test, or it's also called hypergeometric test. And I think Carson already talked about this yesterday. And then you can get a p-value that can help you to know whether it's significant or not. And because when you use gene ontology or other databases to do this type of analysis, you are testing many different biological processes or pathways at the same time. So you also need to do the multiple test adjustment in order to justify your observation. So I want to mention the limitation of this approach. First, you have to define what is differentially expressed, what is not differentially expressed. That means you have to set up a cutoff. For example, people usually use false discovery rate 0.01, let's say, or 0.05 as a cutoff. But the question is, how about the gene right below the cutoff, like 0.011? I mean, is that not useful at all? Probably not, right? But setting this cutoff is very arbitrary. And secondly, after you set the cutoff, for example, for all the remaining genes with a p-value or FDR less than 0.01, you consider them as the same, right? But actually, they're not the same. Some of them may have a 10-fold change, but others may only have a 2-fold change. They're not the same. But if you do this approach, basically, you consider all the significant genes the same. So that is a major limitation. And the gene setting enrichment analysis basically addresses this limitation. It uses a rank list in order to do this. So basically, instead of looking at the overlapping, it uses data from all the genes. And basically, you can rank all the genes from the most downregulated to the most upregulated. And then for a gene set of interest, you can look at the location in the ranked list. If there is no association between the ranked list and the gene set, and you would expect all the genes to be randomly distributed or evenly distributed across the ranked list. But in this case, for example, you see kind of the over-representation of these genes at the top of ranked list. That's why you may think, well, something, there might be an association between this gene set and this ranking. To do that, we use a GSEA statistic, or it's a statistic test derived from the Commongraph Smirnov test. I think Carson already talked about this today, so I won't go into the detail. But anyway, from this, and then you can generate the random simulation, and then you get some random distribution, and then you can get a p-value. Again, if you do this for multiple gene sets, like all the Go categories, all the pathway databases, you need to do the multiple test adjustment. This is better in a way than the over-representation analysis, but still there are certain limitations, like it relies on existing knowledge on the pathways and databases. But of course, I mean, knowledge on the pathway and database and the gene ontology, I mean, those annotations are still limited. For a lot of genes, we don't really know a lot about their functions. And also, it treats one pathway as a separated entity. It ignores the crosstalk between the pathways. But we know, and although you can consider that as a relatively independent unit, they still talk to each other. So as I talked yesterday, every protein is actually linked in the cell system. So that's why the second type of approach is to map your data to the network and then do some network-based analysis. We talked about a few different methods yesterday, and we also mentioned about this pathway interaction databases. And the basis for using this type of analysis is the proteins lie close to each other in the network tend to have similar functions. Yeah, so basically we talked about a few different methods, and so in the WebGestalt we basically implemented two approaches. One is the module-based approach. Basically for each network we pre-compute the modules, and then we treat each module as a gene set, and then you can do the enrichment analysis against those predefined modules. Or you can use a diffusion-based approach, and that will allow you to do gene prioritization. So this is a kind of an overview of the WebGestalt. So the goal is to translate a list of genes of interest into some biological and pathway level understanding. And the system can support 12 organisms. So not only human or mouse, but other model organisms can also be supported. I talked to some of the students in the audience, and I know some of you don't actually work in human, although all the examples today I'm going to give will be in human, but if you work in C. elegans, for example, you will be able to use this resource as well. And we support a lot of different types of gene IDs. For example, whether you use UniPro or whether you use Ensemble to do the proteomics database search, you will be able to use this without worrying about the system don't recognize your ID. And then there are a total of more than 100,000 gene sets, different types of gene sets that can be used to do your analysis, and coming from the gene ontology, from different pathway databases, as I said, from different types of protein-protein interaction networks, or from the gene cancer-derived gene co-expression networks, and also like phenotype-associated gene sets, drug-related gene sets, etc. And we support all three types of analysis, like the overrepresentation analysis, GSEA, and the network topology analysis, which is, basically the diffusion-based analysis. And then the output is very interactive. And this was based on a figure in the 2017 paper. But today what I'm going to demo is 2019. It's not 2019 yet, but we're preparing for that paper. So actually this is my first demo of that new system, and you guys will be the first group of people to know this new system. It's still the beta version, but I think it's quite stable, and you guys should be able to use it now. Yeah, so I put some hands-on next step-by-step guide about the examples we are going to go through today. So you can go to the workshop area, and have everyone got that PDF file or Word file about the steps to do the analysis. So yeah, let's get started. It's a web application, so you don't need to install anything or download anything. You just go use your web browser to go to the right URL, and then you should be able to get started. I would recommend you to use Chrome as a browser because most of our developers are, sorry, I don't know what is causing the, yeah, most of our developers are using the Chrome as a primary web browser for the development, so it's the best tested in that browser. But if you use Safari, or I think there is a guidance at the bottom of this. If you use Google Chrome, Safari, or the latest version of the IE, I think you should be fine. But Chrome is free, so I think anyone can download and use Chrome if possible. So let's go to the WebGestalt website, and you can either just type in the URL you have, the 2019 one, or you just Google WebGestalt and then go to the 2019 beta version, and you should see this interface. And in the first example, I will show you how we can use WebGestalt to understand the list of genes that are associated with colon cancer. It's already pre-configured as ORA sample run. So let's say you click on this ORA sample run, it will automatically fill in all of the parameters and also upload a gene list. So this is a list of genes related to colon cancer. We got this list from another tool developed in the lab called Glad4U. Gene list automatically derived for you. So that one, so basically you can type in anything you are interested in, and then it will give you the list of genes that are related to the concept you are interested in. It could be a disease, could be a biological process, or could be something you are interested in. But anyway, this time we get 487 genes that are related to colon cancer from that tool. So we can go through the parameter setting to just try to understand what this means. So the first one is either it's select organism of interest, because these are the genes from the human, right? So you select Homo sapiens. But if you have other data from other organisms in your future research, you can select the right organism in the future studies. And then select method of interest. We're going to do over-representation analysis because we don't have a rank list. This is the type of situation that you have a list of genes without any statistical metric or anything that you can use to rank them. For example, if you do a clustering and then you get a list of genes just in that cluster, and then there is not really a rank, right? Then you cannot use GSEA, and so we choose the ORA. And we will just use a gene ontology biological process for this basic analysis. But here you can see, in addition to gene ontology, the functional databases related to pathways, network modules, disease-related gene sets, drug-related gene sets, phenotype-related gene sets, and genes in different chromosome locations, or some community-contributed gene sets. And for example, some of you are interested in certain types of studies, like in infectious disease or other diseases, and you may sometimes have a list of genes that you are interested in, and you want to contribute to the community, allow other people to test their data against your gene set, right? This is why we created this community contributed. These are from some labs, they want to share their gene sets to let others to compare their new studies against their gene sets. So let's see gene ontology and the biological process for today. And then the gene ID type, here what we have is gene symbol. But as you can see, we support a lot of different gene IDs, from all the different types of microarray IDs to the Illumina array IDs. And then for proteins, we have the ensemble, RefSeq, UniPro, all these different types of IDs. But this one, particular one we're using is gene symbol. And the reference gene list, in this case, we use a gene, all the protein coding genes in the genome, because this was basically a list come from the literature search, and we don't really know what is the space they use to, we just consider every gene has been, probably have some opportunities to be studied. But in your own research in the future, it's very important to consider what to use as a reference set for enrichment analysis. For example, if you do RNA-seq, it might be safe to use all the genes, because RNA-seq is kind of unbiased. You get access to investigate all of the genes in the genome. But let's say if you use proteomics, specifically, usually you can only get part of the proteomes. A lot of the proteins you didn't identify. So when you say you identify 100 differentially expressed proteins, that's out of the maybe 10,000 proteins you quantified, or even sometimes 8,000 or 3,000, right? So a lot of proteins you didn't have the opportunity to get the statistics, and then you should limit your search space or the background reference space to those proteins you can actually analyze. This is very, very important. I review a lot of papers, and sometimes people just use for example, in a lot of proteomic studies, they use a complete, like all the genes in the proteome as a reference. That is not correct, because that will very likely, you can easily identify, for example, ribosome or those type of highly abundant proteins as enriched. But it's just because those proteins has better chance to be detected, not because they are differentially expressed. Just to be careful in selecting your reference gene list. Upload gene list. Yeah, here, yeah. This one was from, because we clicked on the sample run, it's automatically filled from the, as I said, from the 417 genes from the data, another tool. But in the future, let's say if you have 100 gene samples, you just copy and paste here, or you can go here and you can save this in a text file, and then choose the file, and then you can upload as a file as well. There are two options. Either you can just copy and paste, or you can save that as a file and upload as a text file. But make sure when you upload as a file, your file extension has to be .txt, and then you don't want to have any special characters in your file name. And for the advanced parameter settings, the minimum number of genes in the category, we don't want to look at the categories that have only 1 or 2 genes, because that is not very interesting, right? We also don't want to look at the gene ontology terms with maybe more than 2,000 genes, because those are too broad to be meaningful. And then for the multiple test adjustment, we choose Benjamin Hodgberg correction. So this is one of the most popular, or I recommend just use this method for multiple test adjustment. It's less conservative than the bufferoning, for example. And then for significance level, there are 2 options here. And you can use a FDR cutoff, for example, 0.05 or 0.01. But when you do the first round of analysis, usually I choose the top 10 or top, you can change the number. But so this way you can always get some result back, like you know what are the most enriched term look like. And then, because sometimes if you pick, for example, 0.01, and none of the terms come back as significant, and then you end up with an empty result set, that's not interesting, right? So, and sometimes if your difference is really huge, you end up with thousands of enriched terms, and then you cannot comprehend all those either. So do the top 10 as the first step can give you a sense in how strong the signal is in your data set. And the number of categories visualized in the report. So I think we generate results for all the significant gene sets, but for example, in this case, we are going to visualize the results in a DAG. If the number of gene sets is too big, then for example, if you have 1,000 significant sets, and then the visualization will be too crowded and you won't be able to see anything. So that's why we need to set a cutoff here. And then you want to color the terms in using continuous data, meaning using the significance level in the DAG. You can also choose binary, but usually continuous is a better option. Well, you can download the result. I actually downloaded the result on I can use that as well, but let me try. Yesterday I found sometimes I submit the first time it didn't work and I submit a second time. So I can show you the result I got here. But I hope, yeah, I also got the result. So maybe let's use this. So do most of you get your results back like this? So in this report at the very top, I mean, some of you might have used the old version of the WebGestalt before, but I think this new version is much improved on the user interface. The results are much simpler, much easier to understand and easy to browse. So at the very top, you see the job summary. If you expand this, this basically will remind you about all the parameters you used or, I mean, what you did for this analysis. You can expand and read this, but you can also close it.
I hope today's session was useful, where you got a brief idea about WebGestalt. Dr. Bing Jiang discussed about different type of input ID and how the three methods are different from each other. In conclusions, when we do not have a statistical value with the gene list, we can do ORA, but if we have a statistical values like p value, we can go for GSEA. Lastly, he also described briefly about the job summary, how it looks like and what are the information we can obtain from it. In the next hands-on session, we will learn more about the result visualization, protein-protein interaction modules, pathway based method and network based methods. Thank you.